RNA gene sequencing in identification of etiological agents of COVID-19 associated mucormycosis. So, COVID-19 is caused by SARS-CoV-2 virus. COVID-19 is associated with an extensive range of opportunistic bacterial and fungal infections. Mucormycosis is one of the opportunistic fungal infections associated with COVID-19. Factors favoring growth of mucormycosis in COVID-19 patients are low oxygen, high glucose, ketoacidosis, decreased phagocytic activity of WBCs, and immunosuppression. Mucormycosis is diagnosed by KOH mode and culture on SDA, which is further examined by LPCB. The specificity and sensitivity of KOH and culture are questionable. Hence, we have looked diagnosis at molecular level. Molecular DNA is mucor DNA is extracted from tissue and is subjected to semi-nested PCR with V3 and V4 variable regions of 18S rDNA. The samples negative with semi-nested PCR were done PCR with ITS reagent universal primers. Then all positive samples were sent for Sanger sequencing. My study is a prospective study done from 1st June to 1st August 2021. It consists of sample size of 50 comprising of 40 male and 10 female. The underlying cause of mucormycosis are diabetes, steroid usage, and neutropenia. Almost all the cases were diabetes, either they developed previous history of diabetes or developed de novo diabetes after COVID-19 infection. History of steroid usage was present in 54% of patients. Biopsy samples of included in my study are stress of maxillary sinus, rhinocerebral orbital mucormycosis, mucosal secretions. Excuse. Among 50 samples, 82% were KOH and culture positive, 14% were KOH and KOH positive and culture negative, 4 were KOH negative and culture negative. Molecular diagnosis of mucormycosis. DNA extraction was done according to Romanelli et al. That is published in 2014. Tissue homogenization was done in Falcon's tube. Proteinase K was added. Was incubated with lysis buffer at 75 degrees centigrade for 2 hours. Centrifugation was done with at 12,000 rotations per minute for 15 minutes. Supernatant was precipitated with isopropanol. Then it was given a wash with 70% ethanol and finally diluted to 5200 nanograms per microliter concentration. In semi-nested procedure, the extracted DNA was targeted against V3 and V4 variable regions. That is, in round 1 PCR, we used Z1 and Z2 primers. Amplicon was read at 407 base pairs, which was the target region for round 2 PCR. In round 2 PCR, Z1 and Z3 are the primers. The Amplicon was read at 174 base pairs. The samples which were negative for semi-nested PCR were done PCR with ITS region universal primers. Amplicon size of ITS region varies from 650 to 850 base pairs depending on species. The thermal profile of all PCR cycles are similar except annealing temperatures. The annealing temperature varies from species to species, so primers to primers, that is 60 degrees Celsius for Z1 Z2 primers, ITS primers, and 55 degrees Celsius to Z1 Z2 primers. The PCR product was separated using 1.5% agarose gel electrophoresis and visualized under UV light. This is the gel representation showing lane 1 Z1 Z3 174 base pairs and lane 2 Z1 Z2 at 407 base pairs, and lane 3 we have observed at alpha 1 antitrypsin. It is a local primer, and lane 4 it is the internal transcript spacer gene read at 650 base pairs, and this is the ladder. To identify the species, amplified PCR products were sequenced by using Sanger's method and often sequence were visualized using Flinch TV software, and each sequence was aligned using BLAST. My results, demographic data. Mean age of my study was 46.9 plus or minus 10.5 years. Minimum age was 25 years and maximum age was 70 years. Of these, 4 died and 46 survived. Mucormycosis clinically presented as rhino-orbital mucormycosis and pulmonary mucormycosis in my study. 50 samples were subjected to semi-nested PCR. Of the 33 were positive in round 1 and round 2. Further on sequencing, they were identified as mucor. The negative samples were subjected to ITS universal PCR, 3 samples were positive. On sequencing, they were identified as non-mucor fungal infections. The type of fungal infections identified PCR were 46% were rhizopus and 14% were sporadinella, 6% were amylomyces and 4% were aspergillus and 2% were candida. And of the mucus species, the most of the species were found were Rhizopus microsporus. That is, 12 species were Rhizopus microsporus, followed by 7 species as Rhizopus stolonifera, and least is Amylomyces rosei, that is 3 species. And non-mucor species were identified as Aspergillus flavus and Candida albicans. The species identified in culture as Rhizopus species were 23. These 23 species on PCR were identified as Rhizopus microsporus, Rhizopus stolonifera, Rhizopus erices, Peridinal ambulata, Amylomyces rosei. And the species identified as mucor or the growth which is seen as mucor was identified as Rhizopus microsporus, Peridinal ambulata, Amylomyces rosei in PCR. And the species like Aspergillus and Candida were identified as Aspergillus flavus and Candida albicans in PCR. The culture negative samples were identified as Rhizopus erices, Rhizopus microsporus, Rhizopus stoloniferus, Peridinal ambulata. PCR positive in correlation with standard diagnosis were 36 samples, out of which 28 were KVH positive and culture positive. 5 samples were KVH positive and culture negative, and 1 was KVH negative and culture positive, and 2 samples were KVH negative and culture negative. My sensitivity was 75%, specificity 100%, negative likelihood ratio is 0.25, positive predictive value is 100%, negative predictive value is 99.8%, my accuracy is 99.8%. My discussion: the most common mucor species identified through PCR sequencing was Rhizopus microsporus, which was in correlation with recent study by Crick Walter et al. 2020. Similarly, Sporadinella umbellata which was known as entomophilic pathogen, was identified in 6 samples. According to a recent study, for the first time, Sporadinella umbellata was diagnosed in the clinical samples of zygomyces patients in Iran. That is according to Barisa et al. Published in 2020. Using 18S rDNA Z primers of 33 cases, 3 cases showed Amylomyces rosei or Rhizopus erices. Amylomyces rosei is not a novel species, but may be mutant variant of Rhizopus erices. In this study, molecular diagnosis of mucor through PCR showed 75% sensitivity and 100% specificity, which in correlation with the existing literature, Bialek et al. Published in 2005 and Gardi et al. Published in 2018. The samples negative for PCR cannot be considered as true negatives, can be due to lower concentration of fungal DNA in the total DNA extracted. Conclusion. My conclusion is targeting 18S rDNA regions were suitable biomarkers for the development of rapid, simple and reliable diagnostic. PCR based assay for mucormycosis and our study insights Rhizopus microsporus has a common causative agent. Take home message. Molecular methods are more promising, less time consuming and high sensitivity in identifying mucor species on fresh tissue samples than culture methods. These are my